Tony is from Japan, from the Earth Life Science Institute. He's also affiliated with the Blue Marble Space Institute of Science. Tony, please.
Okay, thank you, Doron. Thank you, everyone, for giving me an opportunity to share about some of my research that I've been doing. I'm a first-time attendee, so I'm very glad to participate here. And so the title of my talk is DNA liquid crystal coacervates and other phase-separated systems as primitive compartments. As Doron mentioned, I'm a researcher at ELSI, the Earth Life Science Institute at Tokyo Institute of Technology. And a little bit of my background, actually. I actually started my research career at Caltech. I studied proteins and then a little bit about lipids. I then moved on to Harvard University, where I studied RNA sequencing, and I also studied non-enzymatic RNA polymerization with Jack Shostak. And I was very happy to hear mention my former colleague, Noam Prius, also by one of the former speakers. So it's nice to see that there's like the global network of researchers coming around again. And so studying, nucleic acids, lipids, proteins, these are all important biomolecules. And it got me thinking and very interested in how exactly these biomolecules originally emerged and assembled and evolved on early Earth. That led me to my current position at ELSI, where I've been researching various projects related to origins of life, astrobiology, prebiotic chemistry since 2017. So let me start the research portion of my talk here. So this is a little bit of background. The modern cell is very complex. For those of you studying biology, you might be studying a few parts of this. And many of the researchers studying the cell are very interested in how each particular part works. And we've learned a lot about how the cell works in the last 10, 20, 50 years. But one of the big unanswered questions is how exactly the cell initially emerged and assembled in the first place. And it's believed that a type of primitive compartment, also a protocell, some people call it, may have existed before the emergence of the initial cells that contain maybe some type of boundary, some types of genetic or replicating material within the boundary. And so in Origins of Life, one of the big mysteries is to understand how exactly prebiotic chemistry, the chemistries available on primitive Earth, could have assembled into the first compartments on Earth, and how and whether these compartments could have evolved into modern cells. And so this is a little bit about how some researchers, including myself, approach some of the questions that we're interested in answering in the Origins of Life research field. In particular, I'm interested in this primitive compartment, and compartments are very crucial to Origins of Life because they perform a number of essential functions, such as concentrating and segregating reactants, promoting chemical reactions, and even protection of molecules from degradation. Recently, there has been proposed, well, not recently, but maybe almost 100 years ago, but recently it has gained more interest, is one mechanism of compartment formation that could have been possible on early Earth, and that's called liquid-liquid phase separation. Again, it was proposed maybe over about 100 years ago by Oparin and Haldane, and just very recently, more researchers have begun to study phase separation in biological systems. In particular, there are a number of membraneless organelles in modern biological systems, and so phase separation is a process that forms membrane-free droplets that forms these small types of compartments that might have performed a number of relevant functions on early Earth. So one of the model systems that is studied in the lab is complex coacervates, and these are small droplets formed from binding, mostly electrostatic, but some hydrophobic or other non-electrostatic interactions, binding of oppositely charged polyions, such as a cationic peptide or an anionic nucleic acid. And here, what happens is these droplets form that are very concentrated in the peptides and the nucleic acids, and the surrounding solution is very dilute. Here's a microscopy image of an ATP and a polylysine coacervate droplet after mixing. So these coacervates have been shown by a number of research groups to perform potentially relevant functions on early Earth, such as concentration of biomolecules such as nucleotides or nucleic acids. Some of these coacervates can grow and divide given external stimuli such as electric fields or shear mix forces, mixing, agitation, vortexing. Some coacervates can also exist as multi-phase compartments, basically coacervates with different compartments that have different chemical structures or affinities. And finally, coacervates also have been shown to scaffold the assembly of membranes around themselves, suggesting the possibility that coacervate compartments could have pre-existed membrane-bound vesicle-like protocells, which may then have pre-existed membrane-bound modern cells. And so this is one way by which coacervates can increase or have been shown to increase in structural complexity through membrane assembly. And so we're interested in studying coacervates and also observing other mechanisms by which these coacervates could increase in structural complexity. And so this led us to study coacervates in the context of another self-assembled system, DNA liquid crystals. So this is a system that is very interest of mostly, perhaps you've heard of them in television screens or other engineering applications, but it is also perhaps relevant to prebiotic chemistries and also modern biologies. And so DNA liquid crystals can form when short DNA duplexes, shown here, can aggregate end to end. They form these small, these short duplexes will aggregate and stack blunt end through their blunt end interactions, forming these long stiff rods. And these stiff rods will then aggregate to form these bundles of rods that form liquid crystal phases. Shown here is a polarization microscopy image, which detects the supramolecular chirality of a system. And so through polarization microscopy, we can identify whether a system has liquid crystal character or not. So in this system, we studied a short DNA duplex shown here. It's a 12-mer. That has been shown to form liquid crystals by itself. And we wanted to introduce a cationic peptide, in this case, very long chain polylysine, which is a positively charged peptide in neutral solution. And we wanted to see whether mixing these peptides and this DNA could form co-assemblies of liquid crystals and coacervates. And indeed, based on what we believe to be mostly governed by an end-to-end stacking interaction, the coacervates and the liquid crystals could co-assemble. Seen here on the top is a normal bright field microscopy image showing this droplet structure. On the bottom, we see the polarization microscopy image showing that the droplet structure is co-localized with the liquid crystal structure, suggesting successful co-localization. However, we also studied the conditions by which this assembly was favorable, and we actually noticed that the favorable range of, for example, sodium chloride concentrations is very narrow. At very low salt concentrations, you see precipitates forming. At higher salt concentrations, you see perhaps non-liquid crystal droplets followed by no assembly at all. And we believe this is because the salt concentration, the ions actually provide a shielding effect that at lower concentrations allows the DNA and the peptide to interact more strongly, whereas at higher interactions, the DNA peptide at higher concentrations, the DNA peptide interaction is more weak. And so here at lower salinities, stronger interactions result perhaps in these precipitate forms, whereas at higher interactions, the liquid crystal phase disassembles followed by the coacervate disassembling altogether as well. We next wanted to see exactly whether this end-to-end stacking interaction was the process governing this assembly. And we designed a duplex with a 2-base overhang, TT, and this results in no ability for these short duplex DNA to stack end-to-end. And so we noticed that in all of the salinity conditions we tested, the only structures that formed were non-liquid crystal coacervates, suggesting that because the 2-base overhangs prevented the small duplexes from stacking end-to-end, forming these long rods, that liquid crystal assembly wasn't possible. The next step, we wanted to subject these liquid crystal coacervate structures to other perhaps relevant phenomena on early Earth, in this case, dehydration. And seen here, we subjected a system starting from a precipitate phase to dehydration, simply from drying. And here's a video showing the transition. Essentially what happens is the structures will transition from a precipitate state to a liquid crystal coacervate state, followed by formation of a liquid crystal, formation of a non-liquid crystal coacervate state, followed by disassembly of the coacervate in general. And we believe that this phenomenon is because the dehydration process, this is at room temperature, a very small volume, the dehydration process actually up concentrates the salt concentration, the sodium chloride concentration, so that upon further dehydration, the sodium chloride concentration increases, resulting in disassembly of the structures in similar accordance to what we observed previously when just mixing the solutions at different salinities. And then finally, we wanted to test whether what the response to different heat stimuli of these systems were. And so starting again with a mixed precipitate structure, we heated the system up to 60 degrees, and we observed that the precipitate structure actually transitioned and converted to a liquid crystal coacervate structure. Upon further heating, the liquid crystal structure disassembled, forming just a non-liquid crystal coacervate structure. And then upon decreasing the temperature, we notice at about 40 degrees that there's coexistence between non-liquid crystal coacervates and also liquid crystal coacervate structures. Here, we actually see two different types of liquid crystal coacervate structures. One is the cholesteric phase, which we observed before. Next is a new columnar-like phase, which was not a new structure. And upon further decrease in temperature, we found that the predominant structure was the columnar-looking phase, the columnar-like phase of the liquid crystal coacervate. And so what's interesting to note is that after heat, after annealing, basically after heating and cooling, the structure looks completely different. Whereas before heating and cooling, a precipitate was the preferred structure. Afterwards, a liquid crystal coacervate was the preferred structure. And perhaps this is due to the preferred thermodynamics of the system, basically the system being able to access the most stable conformation after the heating and cooling, the annealing step. And so here's a brief summary. Essentially, we've changed by changing the salt concentration, we can see different structures of liquid crystals and coacervates. And B here is actually showing the system after heating and cooling, and we can access after heating and cooling a number of different types of liquid crystal structures co-localized to coacervate phases. And upon increasing heats and salinities, the bottom row shows what we believe could be happening structurally within the droplets. At first, the columnar phase, we see these long, stiff DNA rods in blue bound to these peptide strands, forming these tight hexagonal bundles. Upon increase in heat and salinity, we see then these bundles start to come apart, forming the N star, the cholesteric liquid crystal coacervate structure. Following this, the liquid crystal structures disassemble, presumably because of disruption of the N10 stacking interaction, and perhaps even more heating and salinity increases results in complete dissociation of the DNA and peptides originally bound together through mostly electrostatic interactions, resulting in disassembly of the entire system. And so for those who are interested in some more details of this study, I encourage you to look at the study which we published last year. This is a study more about the physics of the system. This is a study more about studying the different interaction of the reactions of the system upon subjecting it to different external perturbations like heating, cooling, wetting, drying, or changes in salinity. And if you're also interested, there's some other work that I've published in the past related to membraneless polyester droplets, which of course I'd encourage you to take a look at some of these papers as well, which have been published over the last couple of years if you're interested. And so I'd like to thank all the funding sources here. Most of it is supported by grants from Japan's Society for the Promotion of Science, JSPS. And I would like to thank my collaborators, in particular Tommaso Fraccia on the right, who is an expert in DNA liquid crystals, which I had a great time collaborating with on this project, and also some of my collaborators on other projects at ELSI, Irina, Jim, and Kuhan, which some of you may know. They've been around the origins of life research community. And so I'd like to thank them as well as my other colleagues and collaborators at ELSI, Tokyo Tech, and at various institutes around the world. I thank everyone so much. And if you're not familiar, ELSI is located in Japan. Our goal is to study the origin and evolution of planets, life, and life on Earth. And hopefully, after this pandemic is over, if you're able to, you please visit us, and it'd be great to have you. So thank you very much for your time and attention. I'm happy to take any questions now, or if you don't have an opportunity, please contact me by email anytime.
Thank you, Tony, very much for this very nice talk. We have time for questions.
Yes, so the first one is, are the liquid crystals stable or transient at room temperature?
Yes, so the liquid crystals, we believe, are stable at room temperature as long as none of the other parameters change. For example, if the liquid is evaporated, then the liquid crystals will not be stable. For example, if the temperature changes are too high, liquid crystals would not be stable.
Mm-hmm. And the second question we have is, do you think the coacervation function is dependent on the sequence of DNA that you have employed?
Yes. So in this case, I don't think it's entirely dependent, and it's been shown that generally that DNA or RNA of a specific length may still be able to coacervate as long as the form coacervates, as long as the I guess the cognate, well, the partner peptide is also long enough or has enough charge. However, there is some studies that have shown that different DNA bases, for example, interact more or less strongly with peptides. I think, for example, arginine will interact more strongly with guanine than some of the other bases. And so at very short DNA lengths, at lower concentrations, there may be some sequence dependence. And certainly, even though we can form coacervates, the physical properties of the coacervates may change. For example, changing the DNA sequence could result in different fluidity of the internal structure, or for example, something might be more fluid or less fluid, or more solid-like or less solid-like. So there are some possibilities to tune the structural conditions through sequence modifications.
Mm-hmm.
Tony, I want to ask you a general question about coacervation. Surely 100 years ago, Oparin was a visionary in saying that there must be colloidal particles to start life. That is, that compartmentalization is important. And but if you look at what we usually do, if you listen to some of the lectures about prebiotic evolution, people are very careful about what could form the first entities. If it is peptides, what amino acids were available. Same for polynucleotides and so on. I think that it's important to ask what could lead to coacervation in the first place, because there wasn't DNA or proteins, etc.
Yes, that's a very good question. And first of all, I'd like to set the record straight that the jury is still out whether coacervation is entirely relevant or related to the formation of the first cells. I think it's a reasonable or interesting direction to pursue, but it is not guaranteed that coacervation itself would have been very prevalent on, well, it's not a guarantee that coacervation would have directly led to, for example, cells or protocells. As for what could lead to coacervation, yes, like DNA, peptides, they would not have existed on early Earth. And so here we presented a system which results in complex coacervation, which is a very specific mode of coacervation where these long, oppositely charged polymers can interact and form these coacervates. There's another type of coacervation, which is dissociative coacervation, essentially formation of, for example, aqueous two-phase systems, or other droplets just that are aggregated out from solution. And these droplets can form from differences in, for example, chemical affinities or differences in polarity. And perhaps the first coacervates or membraneless droplets, and coacervation, I think, also is a very poorly defined term, but the first membraneless droplets, if you will, could have formed from, for example, collections of arbitrary polymers that together were insoluble in solution, for example. Some of the work that I worked on before, something like polyesters, although this is a very pure system, we showed a very pure system, which might not have been relevant or plausible. Mixtures of polyesters and other similar types of polymers forming these insoluble aggregates could have formed membraneless droplets as well on earlier. So perhaps that was what the first types of coacervates would have looked like.
Thank you very much. This was a very nice answer. Thanks, Tony. And thank you for coming over, even though not physically. We will find a way to have you here physically.